Welcome to Completely Biology. This is our 6th class on molecular basis of inheritance. We already know that RNA is synthesized from DNA by transcription and there could be different types of RNAs. For translation, 3 types of RNAs are important. That is rRNA, mRNA and tRNA. So what is rRNA? rRNA is the ribosomal RNA as it is the key component of ribosome. Ribosomes, the cell organelles that is known as protein factories, are made up of RNA and protein. And these types of RNAs are known as ribosomal RNA, that is rRNA. And it is the most abundant RNA in the cell. This rRNA is transcribed by RNA polymerase 1. Another important RNA is mRNA, that is messenger RNA. In our previous class, we have already discussed how mRNA is synthesized and it is transformed from pre-mRNA to matured mRNA. These mRNA act as a template stand for translation. The instruction for a particular polypeptide chain present in the base sequence of its nucleotide. It is called genetic code. Suppose this is a mRNA. It is the longest RNA with maximum molecular weight. Suppose this is the base sequence. 3 adjacent bases specify for a particular amino acid. And these triplet bases is called codon. Formation of polypeptide chains occur in ribosome and mRNA gets attached to the ribosome. tRNA brings a particular amino acid according to the codon present on mRNA. So we need to know about tRNA. tRNA, that is transfer RNA, is synthesized like mRNA. But RNA polymerase 3 transcribe it. Suppose this is a pre-tRNA. Just like pre-mRNA, pre-tRNA needs to be transformed into mature tRNA. Then an extra CCA sequence will attach to its 3' end. Suppose this is the tRNA and this is the 5' end and it is the 3' end. So in this position, CCA will be attached. A specific amino acid will be attached here. So it is called amino acid binding site, AA site. Suppose this is a amino acid. After formation, these tRNA become twisted and appears to be clover leaf shaped structure. So the tRNA is now twisted like this way. This is the 5' end and this is the 3' end and this 3' has an extra sequence that is CCA. This is the amino acid binding site, so it has an amino acid. This clover leaf structure results from the base pairing, producing 4 base pair stem. This 4 base pair stem is separated by 4 loops, that is number 1, loop number 2, loop number 3 and loop number 4. Loop number 2 contains 3 anticodon sequence which will pairs with the codon sequence of mRNA by complementary base pairing. This pairing is very important for translating protein. So this is known as anticodon loop. Several nitrogenous bases of tRNA gets modified like pseudouridine, which is denoted by psi. Loop number 4 contains psi and that's why it is known as T psi C loop. This is the site for attaching to ribosome. Another modified nucleotide is dihydroxyuridine, that is DHU, which is present in loop number 1. So loop number 1 is known as DHU loop. This is the binding site of amino acyl synthetase. We will know about this enzyme in translation. So this is a tRNA which is twisted like a clover leaf, and this is the anticodon loop, and it will pairs with the codon sequence of mRNA. So genetic code is very important. It has been proved that triplet of bases codes for a particular amino acid. As we know that RNA has 4 different types of nitrogenous base, and 3 of which form a codon. So a total of 64 codon can be formed. So there could be a total of 64 codon for all amino acids. There are 20 types of amino acids. So for 20 amino acids, 64 codons are there. But 3 codon do not code for any amino acid. That is UAA, UAG and UGA. These 3 codon do not code for any amino acid. These are called stop codon or nonsense codon, as they don't sense for any amino acids. So out of 64 codon, 3 are nonsense. So 61 codon can code for 20 amino acids. This is one of the feature of genetic code. Another important characteristic is start codon, that is AUG. This codon is a start codon. It will start translation. So it is called start codon. So the genetic code has many characteristics. Another important characteristic is universality. That is, a particular codon specify the same amino acids from virus to human. Suppose UCC codes for serine in virus. It will code the same amino acid in any species like human, animal, tree, everywhere. The next important characteristic is unambiguousness. That means one codon specified only one amino acid. That means if this codon UCC codes for serine, it will not code for lysine or leucine or any other amino acid. It will always code serine. This is unambiguousness. Next important characteristic for genetic code is degeneracy. That means one amino acid can be coded by many codon. As we know that 61 codon can codes for 20 amino acids. So it is very obvious that for one amino acid there are more than one codon. For example, serine. This UCC codes for serine. UCA also codes for serine. UCG also codes for serine. UCU also code for serine. So this is called the degeneracy of genetic code. So this is the important characteristics of genetic code. In our next class, we will discuss translation process in details. Thank you.